I think there is awe and majesty in a starry night as well. On the other hand, actually having some understanding of what those stars are telling us about the universe, to me, actually adds to the awe and majesty rather than subtracting from it. And I think the same thing would be true. I think any of us, when we gaze out over the edge of the Grand Canyon, we're struck by, the immensity of the landscape around us. And I'm as awestruck by that as anyone. But again, I think there is something that actually adds to my appreciation, knowing that those beautiful multicolored layers of rocks that I see lining the canyon are actually telling a story about the planet and help us understand the planet's history as a whole.
Hello, this is Robinson Erhardt here with the introduction to Robinson's podcast number 143. And this episode is with Andy Knoll, who is the Fisher Professor of Natural History in the Department of Earth and Planetary Sciences at Harvard University. Andy has done work on a lot of problems out in the field and at a desk, but in general, his work focus straddles the line between the early evolution of life on Earth and how this is entangled with its environmental and geological history. So he's written a number of books on these subjects, but the most recent of them is A Brief History of Earth, 4 billion years in 8 chapters. And in this episode, we stick to the earliest portions of this history. We talk about when, about 4 billion years ago, and how life arose on Earth. And then, just as importantly, how we know about it and what we know about it. Then we turn to some related topics, such as the diversity of microbial life on Earth and how it shaped our environment. And then in just what sense it's fair to say that microbes are our ancestors, because there isn't a linear path between us and them the way that there is with our parents. And then we get into all sorts of other little details. We finish off, though, by shifting gears a bit and discussing the possibility of life on Mars, past, present, and future, because Andy was part of NASA's MER mission to Mars, which examined the soil and geology of Mars' surface through 2 rovers named Spirit and Opportunity. You might recall those from the news in years past. Likes, comments, subscribes, follows, comments, subscribes. All of these are extraordinarily helpful. And if you haven't left a review on Spotify or Apple and you've been enjoying the show, it would be wonderful if you could leave one. So now, without any further ado, I hope you enjoyed this conversation as much as I enjoyed having it with Andy. The prologue to one of your books, Life on a Young Planet, it begins with a poem by Walt Whitman. I've got it pulled up here. And it happens to be one that I really love. I used to have it memorized, but that was a long time ago. So if it's okay with you, I'd like to read it and then talk a bit about how it connects to how you think about your work and science more generally before we get to that work. Okay, great. So the poem, which I'm sure some of our listeners will be familiar with, is When I Heard the Learned Astronomer. When I heard the learned astronomer, when the proofs, the figures were ranged in columns before me, when I was shown the charts and diagrams to add, divide, and measure them, when I sitting heard the astronomer where he lectured with much applause in the lecture room, how soon unaccountable I became tired and sick, till rising and gliding out I wandered off by myself in the mystical moist night air, and from time to time looked up in perfect silence at the stars. So...
Yeah, I think one of the things that whenever you write, you're always trying to understand how you might get into the subject. And at the time, this is 25 years ago, I had been reading Whitman. I like Whitman very much, but this particular poem struck me because as at least as I read it, it seemed to be saying that somehow knowledge gets in the way of understanding the awe and majesty of the natural world around us. I mean, either that or he's simply, whining about a poor lecturer, and I don't think he'd bother to write a poem about that. And so I guess, at least as it struck me, I mean, I think there is awe and majesty in a starry night as well. On the other hand, actually having some understanding of what those stars are telling us about the universe, to me, actually adds to the awe and majesty rather than subtracting from it. And I think the same thing would be true. I think any of us, when we gaze out over the edge of the Grand Canyon, we're struck by, the immensity of the landscape around us. And I'm as awestruck by that as anyone. But again, I think there's something that actually adds to my appreciation, knowing that those beautiful multicolored layers of rocks that I see lining the canyon are actually telling a story about the planet and help us understand the planet's history as a whole. So that's pretty much why I chose to use that. I don't know that everyone would take the same lesson from the poem that I did, but it's what kindled the flames to begin with.
When you say that whenever one writes, they need to figure out how to get into the subject, do you just mean how you're going to introduce it to the audience who might not have any background in it and make it compelling to them?
Yeah, more or less. And I think this is true actually of scientific papers that I write for my peers. The hardest things to write are the first page and the last page. The technical details and in terms of these kind of books, the details of the story that you tell are things that you probably should have down pat. You probably shouldn't be writing in the first place. But there's always this thing: how do I get in? How do I introduce a subject in a way that might explain why I'm writing, how it may relate to, the thoughts and stuff of others. And similarly, at the end, how do you get out of a narrative in a way that somehow summarizes it succinctly to just kind of reinforce the story you've been trying to tell. So that's really it. I think it's, it's not so much a matter of science as just a matter of trying to construct a compelling narrative.
Mm-hmm. Yeah. One thing that what you're saying brings to mind for me is already the Greeks were aware of, in rhetoric, there's logos, pathos, and ethos. And of course, you need to have the material down. But if you're unable to build a connection with your audience, then it's very difficult for them to get them to absorb and have your work resonate with them. So starting out with something that's emotional, like a poem, helps. And that's one of the reasons, or probably one of the reasons, epigraphs have endured so much. But I'm just going to agree with you at first. So I don't think most people see much awe in like a random rock. Someone with geological training like you might, but with just a little understanding of quantum mechanics, just some rock or even a can of Coke becomes this amazing and mysterious object. Yet one other thing that you said in your book, in the introduction, I wrote it down. But does ignorance really outstrip understanding as the preferred route to wonder? And I don't think that by any means you're saying that we shouldn't have wonder. I'm not attributing that to you, but I see there is definitely room for both, and it's the awareness of the ignorance and the visceral experience of wonder that often at least fuels one initial foray into the statistics and diagrams as a way of deeper understanding. But I mean, I witness it often, especially in myself, that one can lose sight of the whole by dwelling too much on the technical dimensions of a field and miss the bigger picture.
Yeah, no, I don't disagree with what you're saying, and it's consistent with what I was hoping to convey in the introduction to that book, that by all means, we don't want to lose our sense of awe and wonder at the world around us, and for many of us, it is that awe and wonder that inspires us to travel down the roads of science to begin with. I just sort of felt that somehow in his poem, Whitman was perhaps shortchanging what the knowledge part can do. Because to me, knowing something about the universe and the stars and that really adds to the awe and wonder. So that maybe ignorance isn't the right word because it touches off too many flames, but I think that the more understanding we have of the world around us, the more we will appreciate its wonders, because they're just as wonderful if you understand them as if you don't.
Absolutely. As we're about to get into, I mean, your books are filled with wonderful pictures. They're particularly wonderful for someone like me who hasn't visited some of the sites like that in Namibia in the background, where if I were just to look at these pictures, I would see striated rocks. But with the aid of microscopes and the accompanying explanations in the text, you realize, wow, these are filled with fossilized remains of creatures very different from us, some of which are still almost, they have relatives that are extant, but in many ways they're quite different from anything we know today. But a nice way, I think, to start off this conversation will be to put some of these massive numbers around the age of the universe, life, the big shifts in climate and chemistry in perspective before we begin to delve into the specifics. First of all, I think the universe is generally thought to be around 13.7 billion years old at present, though this number is subject to change pending the constant refinement of cosmological models. But how old is the Earth, and how long has life been around?
Okay, well, the Earth is about 4.56 billion years old, and we know that because the materials out of which the solar system formed very early in the solar system's history aggregated to form the planets, including the Earth. And some of the materials from that earliest stage of the solar system that did not aggregate into planets became, in the fullness of time, meteorites that struck the Earth. And so we can actually use radioactivity to date those. And so the age of the Earth, being a little more than 4.5 billion years old, is very well established. Now, the hard part with life is that we literally have no rocks from the first 500 million years of Earth history. And the reason for that is that, when we were talking earlier, you get the sense that Earth records its own history in things like sedimentary rocks. On the other hand, while the Earth is writing with one hand, it is erasing with the other through erosion and tectonic processes in this. And as we go back through time, the record of any given time period gets smaller and smaller and smaller until we get to 4 billion years ago, and that's what we have. Now, if you then ask, well, how far back can we trace life? It pretty much is the case that we run out of rocks to look at before we run out of evidence for life. So while we don't know whether life began 4.1, 4.2 billion years ago, who knows? What we do know is that Earth has been a biological planet for most of its history.
You said, you used the phrase writing and erasing in this process of erosion and other geological processes. And it sounds a lot like a Sherlock Holmes story. And as I read about you and your colleagues traveling to various sites, searching for clues, debating alternate hypotheses, practically like metaphorically dusting for fingerprints, it is very much like a mystery novel. I can see you're ready to respond.
Well, I was going to respond only because one of my teachers and colleague and friend for many years was the late Stephen Jay Gould. Great. And one of my favorite essays by Steve is one where he is trying to explain how science actually works. And he did it by comparing two different detectives. One was Sherlock Holmes, for whom everything is deductive and logical, and you come upon the right answer because you've exhausted all the other possibilities. And the other was Peter Wimsey, the detective of Dorothy Sayre, and he recounts Wimsey just sitting in his easy chair, completely befuddled by what he's come across as evidence, and he pours himself a glass of wine, and then in a moment the wine glass goes crashing to the floor and everything makes sense. And Steve's point was that we all would like to think that science works like Sherlock Holmes, but in truth it probably works more like Peter Wimsey, that you just don't understand things and then just something slips into place. It might be something you see on an outcrop, under the microscope, a chance comment somebody else makes, and then it all fits.
Well, this will take us on a bit of a tangent, but I don't know, maybe 10 years ago, read some of Stephen Jay Gould's work and Richard Dawkins, these sorts of people, when I was just learning about this world of evolutionary biology, but he hasn't been around for a while. Naturally, I haven't had the chance to have him on the podcast. Most of what I know about him, his character is from other writers, like he comes up in Daniel Dennett's work often. But I understand that he was a big character and he was a huge figure in the intellectual culture of the academy. What was he like? What was working with him like?
Well, I had a long association with Steve. When I was in graduate school, he was one of my committee members, and he was a young scientist at the time and really making his scientific reputation, which was in many ways to infuse some modern concepts of evolutionary biology into our thinking about the fossil record, although at the same time, he was actually a very gifted historian of science, and in a way really unpacking for the rest of us the thinking that had through time led to current concepts. And in those days he was wonderful. He was always available. He was full of thoughts, generally thoughts that would help you a great deal. And then I went off, taught at Oberlin College for 5 years, and then when I came back to Harvard, for years Steve and I used to teach courses together, and that was great fun too, because he was always very interactive with the students. I think through time, Steve became more concerned about writing than generating new paleontological knowledge, but that's okay. He was a gifted writer, and so we all benefit from his writings. But all in all, I guess I feel privileged to have known him and been a friend and learned from him for 30 years.
Right. I know that one of his books that I read, I don't recall the title, you might know, or maybe he wrote many on this subject, but was on the Burgess Shale, which
right, Wonderful
yes,
Life.
and that I can see immediately how it makes important contributions to the connection of evolutionary biology and the fossil record. But so I think you said that, maybe you didn't say, maybe I'm just recalling this from reading, that life, even though the Earth is about 4.56 billion years ago, life is thought to have been around for about 4 billion years, which is on that order, which is a very long time. And to put that number in perspective, how long have the macroscopic forms of life, so like fish, plants, fungi, been around? And when did the microscopic creatures essentially have to cede ground to them?
Well, the first thing to say is that we still live in a bacterial world. And I think I said it in Life on a Young Planet, that animals are the icing on evolution's cake, but bacteria are the cake. And I think that's true. The world would do just fine without plants and animals. It did so for 3.5 billion years. On the other hand, the world would not wind down very quickly in the absence of bacteria, since they have all sorts of metabolic processes that are not found in more complicated animals like plants and animals. So I'm a rigorous defender of the bacteria. Now, that said, while there's still 30 tons of bacteria on this planet for every ton of animals, about I think the oldest macroscopic animal fossils that we know of are about 575 million years old. So, of that 4 billion year history of life, 85% of it is microbial. And animals are latecomers. Plants and animals both get on land more on the order of 450 to 400 million years ago. So what we consider the modern world, even in its broadest aspects, is a fairly an evolutionary latecomer. And it won't surprise you to know that there are actual environmental correlates to that. Until about 600 million years ago, it seems that the amount of oxygen in the atmosphere and oceans was very low. And for various reasons we could discuss, oxygen starts a trajectory toward its current high level about the same time when we first start seeing animals. And then it really, has the great push through to reach modern levels of oxygen on the same time scale that we start having plants and trees and things that will actually contribute to coal, which is important because when organic matter is made by taking CO2 and water and producing organic matter and oxygen, if you bury the organic matter to form coal, the oxygen has nobody to dance with and can accumulate in the atmosphere. So it's an interesting story in that the history of Earth's environment, or the physical Earth, and the history of life are fairly complexly intertwined through the whole story.
A few things that you say demand, okay, everything you said demands further questions, but two things that come to mind. I think you said that there are 30 tons of microbial life for every ton of animal life. And okay, I'm guessing that if I weigh 170 pounds, there's I don't know how many pounds of microbes living in my gut spread throughout my body. But where is all of this microbial life? Because we can't see it, but it outweighs us by 30 times. So is it just if I look out my window, I see all of these surfaces, the air, the soil, if I just kind of scoop it all up and sift out the microbes, that's where that's where it has to be. But
yeah. This an interesting team at the Weizmann Institute made a quantitative estimate of how much biomass is allotted to each of the major groups of life a couple of years ago, and it's probably not far off. In that estimate, in fact, more than half of the living biomass is in plants, although that's kind of cheating because a tree, wood turns out to be made of non-living tissues. So, 90% of a pine tree outside your door is non-living. But that aside, bacteria occur throughout the oceans. They occur in sediments. They occur in soils. They occur in the subsurface of the Earth. There's a fairly robust subsurface microbial biota, and so when you add it all up, they're all over the place. And you're right that by weight you are mostly a human, but by cell number or individuals or cell number, you are probably mostly bacteria.
Well, the other thing that I wanted to touch on is you said that things would wind down very quickly without bacteria. And just historically speaking, it's kind of amazing that for thousands of years of biology, though we might want to say that biology started with Aristotle, so 2,000 years and some change, it's amazing that biologists had to attempt to make sense of the circle of life, to speak figuratively, without the faintest clue of the most crucial part of the ecosystem, because they had no idea that these things were around. But I think this will come up more if we have a chance to touch on how your work connects with astrobiology. But the Earth was not, as you've already indicated, at its conception endowed with the atmosphere that we currently enjoy, and rather it was fashioned over time by geological and importantly biological processes. So just starting again, putting us in perspective, what was the Earth like 4 billion years ago when around that time the first life arose?
Okay, well, one of the things we've known from the chemists who have done prebiotic chemistry experiments over the years is that none of the experiments that are designed to generate from physical precursors, physical precursors, the building blocks of life work if there's oxygen around. It just doesn't happen. And so even just from that experimental perspective, it suggests that the early Earth would have been oxygen poor. And certainly there are chemical signatures of the redox state of the atmosphere and oceans, which are encrypted into sedimentary rocks. And they agree that on the early Earth, there was essentially no free oxygen gas. And also, 4 billion years ago, the Earth is increasingly thought to have been more or less a water world. That is, that if you had looked at the Earth from, some satellite platform, you would have seen a global ocean with volcanoes sticking up here and there, and maybe a little bit of things that are made of the same kind of crust as continents. But it was a very different world, both physically and chemically.
Well, I would like to get back a little later to this idea that with oxygen in the atmosphere, the precursors of life cannot form, because if we're going to talk about early life, we need to talk about how early life formed, but we'll get to that in a little bit. First, I just wanted to ask one last context setting sort of question. And just again, to highlight how little most people, myself absolutely included in this, probably know about the period between now and even hundreds of millions of years ago. I learned from looking at your book that the first dinosaurs existed before the first flowers, which is certainly not the chronology I would have expected. So are there any other majorly surprising cutoffs or orderings in distant evolution that come to mind just offhand?
Yeah, I don't know that there's, at least to me, the ordering isn't too surprising, and I'm not too surprised that dinosaurs could have existed before flowering plants, because the first land vertebrate animals, first amphibians, came ashore about 360 million years ago, something like that. And so even dinosaurs reflect an antecedent more than 100 million years of terrestrial vertebrate evolution. I think what is interesting is the role that, again, physical processes play in shaping the evolutionary trajectory, because as many of your listeners may know, the reason we don't have dinosaurs today, if you don't count birds, which are actually genealogically dinosaurs, dinosaurs were wiped out by a meteorite that pummeled the Earth 66 million years ago. And our familiar world of mammals really only took shape in the aftermath of that. Animals diversified mightily once dinosaurs were out of the picture. So again, it's very hard to get away when you look at the history of life without thinking about the history of the physical world. And it's equally hard to explain the history of the physical world without considering life.
Well, I have used the word perspective a seemingly inordinate amount of times in this conversation already, but when you're dealing with these timescales, it's crucial. Still, another sort of perspective that I'd like to keep in mind as we keep going is that science is an activity rather than some metaphorical compendium of facts. So I'd really like to talk, or keep in the back of our conversation, about the acquisition and discovery you've been part of the information we're getting into. So that being said, let's get into the earliest of life on Earth, and what specific fossiliferous, a new word I learned. What specific fossiliferous site contains what have been dated to be the earliest fossilized remains of life?
Yeah, life can leave its calling card in the rock record in several different ways. And I think most people would agree that some of the oldest little altered rocks that we can look at are about 3.5 billion years old. There's some of them in South Africa, there's some of them in Australia. And those contain several different calling cards for life. One is if you go to a place like the Bahamas today, you'll find in some areas along the coastline, there are just these carpets of microbes, mostly a group of photosynthetic bacteria called cyanobacteria. And these microbial mats can actually trap and bind sediments and build three-dimensional structures called stromatolites that get preserved in the rock record. And in 3.5 billion-year-old rocks from Western Australia, in some of the earliest carbonate or limestone-like rocks that we know of, there are stromatolites. And we've done work with some Australian colleagues that make, I think, a compelling case that these did require microbial communities to make. At the same time, there are chemical records of life. So, for example, photosynthetic organisms, when they take CO2 and fix it into sugars, it turns out that carbon as an element comes in three flavors. About 99% of all the carbon atoms on this planet have 6 protons and 6 neutrons. So it's called carbon-12, its molecular weight. About 1% has an extra neutron, carbon-13, and that's stable. And then a couple parts per trillion are carbon-14, which is radioactive and decays on a time scale of thousands of years. So the reason I bring that up is that when a plant, or a cyanobacterium for that matter, takes in CO2, it preferentially incorporates carbon dioxide that has the lighter isotope of carbon, carbon-12. And by using an instrument called a mass spectrometer, you can actually calculate the C13 to C12 ratio. And in modern environments, if you look at, say, limestones accumulating in the Bahamas and organic matter in the same sediments, they differ by about 25 parts per thousand. And when you go back in time, that persists, and it persists all the way back into these 3.5 billion-year-old rocks. So we think that's strong evidence that there was a biological carbon cycle at the time. I could have told a similar story about sulfur, that there was a biological sulfur cycle. And then finally, there's the question of actual microfossils. And I've spent many years working on microfossils in sort of Earth's middle age, the so-called Proterozoic eon. And they're abundant, they're well preserved, they're interpretable. But what you see in these 3.5 billion-year-old rocks are just very simple spheres. And it is challenging to interpret them. So there's a lot that we don't understand about life 3.5 billion years ago, but we think A, it was there, B, there were biological carbon and sulfur cycles, and C, the ecosystems worked without oxygen.
Okay, again, a number of things. Is the idea that these stromatolites in any of these given sites were formed by masses of cyanobacteria or by a different type of organism that just happened to form stromatolites?
That's an important point. We don't know that these oldest stromatolites were made by cyanobacteria. I think for Earth's middle age and going up to the present, because stromatolites still form in some areas, cyanobacteria are the major architects. But it's possible that some other kind of bacterium, in fact, something that may no longer exist, was important 3.5 billion years ago.
And then you already indicated this, but there is controversy over some of these findings. And what I find myself wondering is, what are the sorts of alternatives that could make sense of the simultaneous presence of stromatolites, the ratio of carbon-13, the sulfurous evidence, the difficult to interpret potential microfossils that are spherical in nature. What compelling evidence or what compelling alternatives are there that could account for this?
Well, yeah, I think that it is not controversial to state that the geologic record records life at 3.5 billion years ago. And I think it's from there that it gets more difficult. Some people will say, oh, there must have been cyanobacteria, but I think that's an overinterpretation. We do know that there are physical processes that can produce this variation in carbon isotopes that we see. In fact, the most famous of all origin of life experiments, the first one by Stanley Miller in 1953, those reactions actually what's called fractionate carbon isotopes. They actually result in products that are depleted in carbon-13. But if you look at the details, it's very hard to take those kind of physical processes and account for the record as a whole that we see at that time. So I think that the broad statement that there was a biological carbon cycle is well supported. Also, when you look at stromatolites, yeah, there are physical processes in which basically the minerals of limestone simply precipitate and can form layers that way. But again, there are details that can differentiate between microbial mats and physical processes. And even at 3.5 billion years ago, we see some of this telltale textural evidence that I think really requires that there was biological processes. With actual microfossils, it's more difficult. We actually wrote a paper some years ago where one of our graduate students in the department had been doing work on about 800 million year old rocks from northwestern Canada. And he brought over just to look at someday some what's called thin sections, paper thin slices of rock glued to a microscope slide, and you look at them under the microscope. And much of what we were looking at was simply the kind of textures that you see in rocks of this age all over the world. But there were little veins through which hydrothermal fluids had percolated. And in those veins, there was actually some movement, transport of organic material, which then spontaneously condensed into 10 or 20 micron spheres. Looks for all the world like what you see in these 3.5 billion year old rocks. And we know from independent evidence that 3.5 billion years ago, most sediments were shot through with hydrothermal fluids at some point in their existence. So again, we can argue the details, and a lot remains unknown about these things. But I think the overall statement that life was there and it didn't use oxygen is, I think that's pretty compelling at this point.
Actually, I didn't want to cut you off, but I have it on very good authority from a few reliable canonical texts that life is in fact only about 6,000 years old. So I don't think that we should overlook these highly reliable sources, but we will for the purposes of this conversation. How I think you mentioned this, or to be clear, that was a joke, but
yes,
okay.
That's why I'm still on the line.
Well, when I talk to astrophysicists, I find myself often making jokes about aliens, and I occasionally get worried that they think that I'm actually I mean, it's not that it isn't a topic that shouldn't be discussed. We'll get to astrobiology. It's important, but it needs to be needs to be treated seriously, I suppose, is what I wanted to say. But you already mentioned a little bit about how these rocks are dated, but carbon dating hasn't really come up on the show before. And I think it might be interesting to hear about just how we know, for instance, that these sites, the rocks there, are 3.5 billion years old.
Um, it all has to do with radioactivity, discovered in 1899 by Becquerel, something like this. Um, and in radioactivity, there are some forms of elements that are radioactive, which means that on various time scales, they spontaneously transform into other elements. They might shoot off an electron or something like this, but they do actually they're actually have finite lifetimes, and they transform into something else that can be stable. And it turns out that nearly all isotopes known on Earth are radioactive. The stable ones, the ones that make up the world around us for the most part, are relatively few. Now, most of these radioactive elements have very short so-called half-lives, and so they don't last very long. But some of them have very long half-lives, and the most important one is uranium, where two different radioactive isotopes of uranium break down to form stable lead. And we can measure the rate at which that happens in the laboratory. So we now have a natural chronometer. And there's a particular mineral that's beloved by geologists called zircon. It's what, if you ever have cubic zirconium jewelry, we're talking about zircons. Zircons are these incredibly hard minerals. They form by the same processes that make granite, for example. And what's interesting about them is that the crystal lattice of a zircon as it forms can incorporate uranium, but it cannot incorporate lead. So any lead that you find in an ancient zircon had to get there by the radioactive breakdown of uranium. And because of that, we can date zircons rather precisely. And what one of the things that's interesting about that is I said earlier, there are no rocks that are older than 4 billion years on Earth, but there are some zircons that appear as sand grains in younger sandstones that go back to 4.4 billion years. So it is, again, just to recapitulate, it is the process of radioactivity. It is the ability to measure the rate at which different isotopes break down into stable daughter products. And it is then the geologic field work that allows us to go out and date rocks. So, for example, if you look at the rocks behind me, near the base of that cliff down by the river in this region, there are volcanic ash beds. And those volcanic ash beds actually have zircons in them. And the zircons are something like 546 plus or minus 1 million years old. So that gives us a precise datum, if you will, that helps us to calibrate the rock record. And whenever you see a geologic timescale, it's the result of thousands of geologists over many decades going out, finding volcanic rocks in places that allow us to correlate them with fossils or climatic events, and we build up the timescale from that.
If there are no rocks that are 4.5 billion years old on Earth, though we do have some zircons, why, if the rocks that we do have and have found good evidence for life in them, even if the nature of that life is still a bit questionable, why is it reasonable on this basis to conjecture that life originated 500 million years earlier, even though, of course, at those timescales it's fuzzy? But why 500 million years?
Yeah, there are some so-called metamorphic rocks, that is, rocks that have been subjected to originally sedimentary rocks that have been subjected to heat and pressure. And rocks back to at least 3.9 billion years old do show evidence of this carbon isotopic signature that we see elsewhere. So it's only one line of evidence. It's nicer to have multiple ones, but we basically seen nothing in the preserved rock record that is easily associated with an abiological world. So, if you tell me, well, it was really 3.9 billion years ago, fine. I mean, 4 billion years is a number that's brooded about because it's about the biggest, or the simplest large number that you can associate with this. But yeah, sure, there's a fair amount of uncertainty. I think most models for the evolution of the Earth's surface suggest that there would have been liquid water well before 4 billion years. And so, the origin of life could have taken place very early. We just don't know. One other thing about the origin of life and life on Earth that I think bears thinking about is that when people think about life in the universe, they commonly think about, is it likely or unlikely that life will begin on a given planet or moon? And for all we know, it may actually be fairly simple. I think it's worth at least considering that what is potentially unusual about Earth is not that life began here, but that it's persisted for 4 billion years. That is not a given for any planetary system, and it may, it's certainly what led to technological humans in the long run, and it may be something that is much less widespread than the origin of life on different planets.
Now, my knowledge on the scholarship of life's immediate origins is pretty much limited to some of Richard Dawkins' books, in which he describes a hypothetical process in the primordial ooze of this world in which molecules, through a process of natural selection, become increasingly complex and develop primitive self-replication abilities. And then these self-replicating proto-creatures then develop modes of protection, so primitive cell walls, for instance. And on this goes until something like the early creatures we've just been discussing arose. So how closely does this picture I just described correspond to the origins of life on our planet as you see it? Of course, not entertaining in this case the panspermia hypothesis, and because you already mentioned this a little when you said that the early chemistry wouldn't have happened with oxygen in the environment.
There's actually a very rigorous and vigorous laboratory research effort on so-called prebiotic chemistry, and in a sense, it is, as you've described, a perspective that goes back to the Miller-Urey experiment, which really kicked this whole chapter in scholarship off. And that is what Stanley Miller did, was basically took ammonia, water, methane, missing something, CO2, and put them in a flask and ran sparks through them to simulate lightning passing through the early atmosphere. And, after this was going for a fairly short time period, he started noticing brown gook accumulating on the sides of his beakers. And when he analyzed that, there were a number of amino acids that had been created. So we know through not only this, but a large body of work under various conditions since then, that it is fairly straightforward to make the basic building blocks of life from simple inorganic precursors under plausible planetary conditions. So you can make amino acids, you can make sugars, you can make lipids. And in fact, people have now succeeded in even making, the materials that make up DNA and things like this. So getting those precursors is fairly simple. Now, it gets more challenging when you start to link them together into functional entities that actually interact with one another, so that, when you replicate your own DNA, you need proteins to do that work. And yet you need nucleic acids to code for the proteins that are doing the work. So how do we get nucleic acids and proteins to work together? And that's still something where there are a lot of unknowns. But one of the things that's kind of fun is an idea that's been around since the 1970s, but seems to have a new life, is that many proteins actually function with the help of what are called cofactors. Some of the vitamins are cofactors, for example. And many of those cofactors actually have a chemistry that it resembles that of, DNA or RNA. And so some people think, and I think it's, you can't prove it, but it's not unreasonable, is that early on, you do, as Dawkins described in your statements, have nucleic acids that can self-replicate. And we do know now that some nucleic acids can actually function like enzymes. We usually think of proteins as enzymes, the things that actually do the work in the cell. But some nucleic acids can do that. And through time, we seem to have, it may be that the intermediate in leading to the world of proteins was some of these cofactors that, maybe that the amino acids of the proteins were originally there to stabilize the cofactors and then became the most important part of it. And so all of these things are possible. Progress is being made on a year-by-year basis yet, but I think getting that interaction between proteins and nucleic acids that is the basis of modern life, we're not quite there yet. As far as environments, Darwin talked about a warm little pond in a letter from the 1870s, and more people are looking to lakes because lakes can concentrate these organic materials. Lakes can dry up and re-wet, which actually facilitates forming polymers now too. So we actually live at a moment in the history of thinking about the origin of life where there are insights coming from various perspectives, ranging from Earth history to experimental chemistry to insights from molecular biology that are, slowly putting things together. We know a lot more than we did when I was a boy, and I suspect that we'll know a lot more when the boys born today and the girls born today are my age.
Well, I personally prefer another theory, one that I think is much more elegant, parsimonious, beautiful, and that's spontaneous generation, where if you have a bale of hay in just the right barn, eventually mice will spontaneously appear. I much prefer that, but I do understand that it has fallen out of favor. And you've just pointed to a number of theoretical and conceptual tools, I mean, experiments, chemistry, etc., that bridge this gap between the period in which we know that there was no life and these early fossil records. So we have an understanding of how the basic building blocks of life could have formed. Beyond this, are there any sorts of structures or features of the early proto-life that we have a pretty good idea of how they might have formed? So, for instance, I have the understanding that maybe a primitive light sensing mechanism, we can understand how that might have formed, or something like flagella, little ways that tiny creatures can propel themselves about. Are there structures like these that we can just see how they happen through natural selection from these basic or slightly more complex than the basic building blocks?
It's fairly complicated and fairly tough. And I should say, before I get into it, with regard to spontaneous generation, several months ago, I took part in a celebration of Louis Pasteur's 200th birthday at the École Normale Supérieure in Paris. And I have to say that old Pasteur was pretty hard on spontaneous generation. So,
Yeah, well, I can't blame him. I do think it's fun. It's fun to think about. I mean it's fun to try to put your head in other people's heads when they didn't have the science that we have.
oh, sure. Yeah, no. I think in the 1600s, that made as much sense as anything. And again, it was, you talked earlier about most of the history of thinking about life was a history that didn't include bacteria. And really, you know, I think it is because uh, Pasteur was one of the early people to appreciate what bacteria are and what they're doing that allowed him to have these experimental insights with regard to spontaneous generation. But be that as it may, there are certain things that we have some confidence in. So, for example, it is possible to make lipids by non-biological processes. And if you look at, there's a whole class of meteorites called carbonaceous chondrites that can have several percent organic matter in them. And that includes lipids, and they're made,
Fatty meteors.
abiologically. Nice things about
Fatty
pardon?
meteors.
Yeah, basically. And if one of the things that we know is if you have lipids in water and you splash the water, they will spontaneously form spherical vesicles. And when you put them in, let's say, a beaker with, amino acids or polymers of amino acids, peptides, and then let it dry out and let it wet again several times, you'll find out that some of those peptides are now actually inside the vesicles. So very simply, the most basic overall features of a cell, which is to have a protective membrane on the outside and the actual chemical work of the cell on the inside, that all makes sense. Precisely how that happened, what the course that takes us from there to a simple bacterium is, there's a lot to learn. And then, when it gets to more complicated things, and a flagellum is actually a fairly complex thing. There's lots of proteins and everything in it. We get some insights from comparative biology. And so, we know, for example, that the mitochondria in your cells originated as free-living bacteria that essentially became symbionts inside a host cell. We know that the chloroplasts of an oak tree started as originally free-living cyanobacteria that were incorporated and essentially reduced to metabolic slavery within a host. So there are things that we know, and I think once we get into the last common ancestor of all extant life, which is already a complex organism, from there on, I think we can make reasonable insights from, comparative biology, from molecular biology, that sort of thing. It's getting from the Miller-Urey experiment to the last common ancestor that is exciting, and we're learning a lot, but there's still a lot to learn.
This question of the last common ancestor, though, is I think a much more complex question than people realize. In the case of humans, we understand that there is, I don't remember what the technical term for her is, but we all have a common mother ancestor sometime back in Africa. I don't know if it's a couple 100,000 years ago, something on this order. Maybe you want to correct me there, but
yeah, it's something called mitochondrial
yes,
Eve.
exactly.
That suggests that all of our mitochondria and are descended from a single woman, but that's not quite
Right,
the same
right.
concept
That's what I
as
was going to
this.
get to. So am I right that it would not nearly be so simple as drawing a straight line from these early organisms to humans in a tree of life diagram? Because if I am right, I think it would be interesting to touch just briefly on the notion of like horizontal transfer, or otherwise how we might be thought of as chimeras or compositions, because even the mitochondria that you just mentioned indicate that this process is not entirely linear.
Yeah. No, those are good questions. And I think when I say the last common ancestor of, and it can be of humans, it can be of mammals, it can be, you name it. That's really a phylogenetic concept, which is to say that, through a combination of anatomy, morphology, and molecular biology, we are building an ever more refined sense of the genealogical or evolutionary relationships among extant organisms. And if we have that, if we know, the evolutionary relationships of among all mammals, for example, we can make some reasonable insights or with some, reasonable thoughts about features that the last common ancestor of all mammals might have had. We may not know it in detail, but we would suggest that it had hair, it had mammary glands, it had certain kind of teeth, certain kind of bone structures, that sort of thing. And so that's really what we're talking about. And when we talk about the last common ancestor of all cells, at the very least, it had proteins, it had DNA, it had a genome, it had lipids, it had various types of physiological pathways. And all that really comes from looking at the features of its descendants and then sort of logically going down through the tree to see which ones must have been there already at the basal node of that tree. So that's I think what we're talking about.
I'll just put my own spin on what I'm getting at is that not only do branches on the tree of life split, but they also converge. So that mitochondria have their own DNA and had an independent evolution at some point from humans, and then at some point they became encapsulated in our cells, and a symbiotic relationship was born. So that's why what I mean. It's
and so again, mitochondria are a good example. We know because we can look at their sequence structure of their DNA that the ancestors of mitochondria belong to a group called the alpha proteobacteria. And so if you draw a tree of alpha proteobacteria, one of those branches is the mitochondria. And that allows us to make some inferences about what the ancestor of that mitochondrion might have looked like. And as far as we know, if we then look at the tree of eukaryotic life, that is, organisms that have membrane-bounded nuclei, the last common ancestor of all extant eukaryotic organisms had mitochondria. So we can argue about how they got there, and there's a lot of debate about some of those details. But I think we do understand that both at the level of organelles and at the level of individual genes, there is horizontal gene transfer. And it particularly means you have to be cautious when you make trees of things like bacteria, because they have fairly rampant horizontal gene transfer. But it also makes a picture that can become quite compelling. If you look, for example, at mutations that convey defenses against antibiotics, we can see how those move from one bacterial group to another, much to the chagrin of the medical community. But again, if you recognize that these things occur, there are various ways in which you can dive in and try to get a sense of how they've occurred, when they've occurred, what the consequences are for the groups that they now invade, if you will. And that's a very exciting field.
A few thoughts. One are this whole discussion of horizontal transfer and mergings of the tree of life and branchings. It is helpful, I think, in these instances to take the gene's eye view of evolution that Dawkins proposed, where you think of as life as the proliferation of genes rather than organisms or species. But also, we're now players in the game of horizontal transfer with uh, technologies like CRISPR, where we're moving tomato genes to another creature. And then the last thing that I wanted to say is that although with something like mitochondria, this takes place on, well, maybe again, this is a place to correct me, on geological timescales. It was probably a very long time. It took a very long time for mitochondria to become part of our cells. But this is an interesting place for contemporary research as well. My understanding is that within entomology, there are a lot of cases in which we see different organisms living together symbiotically in a way that could presage a potential merging of the species. So one instance from myrmecology that comes to mind is these ants that sort of ferry around aphids and eat aphid poop, basically. The nectar, or I don't recall what the term is for what they secrete, but it's been hypothesized by some myrmecologists that over the course of, I don't know, thousands or millions of years, they might merge.
Yeah, again, you touch on an important point in a very active field of research now, that the idea that animals are completely separate from microbes simply isn't true. As we talked earlier, you have probably more bacterial cells in your body than you do human cells, and at least some of those bacteria in your intestine play an important role in helping you to digest food. They have some regulatory capacity to them, and there's a very active research program on both the functionality and evolution of these symbioses. I think probably every animal has microbial symbionts of some type. It also has a lot of, many of the bacteria in your gut, probably doesn't matter whether they're there or not, and a very few of them are harmful. But I think it is the normal state of animal life to have some intimate relationships with bacteria, without which the animal's life would be difficult, if not impossible. The same thing is true of plants and bacteria, plants and fungi. We're now understanding that the biological world is much more integrated than maybe was thought in the past. And the more we learn, the more remarkable that story becomes.
Yeah. Before I respond to what you just immediately said, I'll add, I started this podcast a few years too late because I would have loved to have E.O. Wilson on the show. The Insect Societies is one of the most thought-provoking books I've ever read, and hopefully there will be many entomologists and myrmecologists on the show in the future. But the last thing that you said was that the biological world is integrated, and while it's integrated, it's also extremely diverse and continuing to get diverser. But returning now to our thread of early life, what are some different ways in which these earliest life forms made their livings? I mean, we're most familiar with creatures that eat and breathe, and then others that make energy out of sunlight. But what are some of the other options? I saw that there were iron breathing bacteria at some point.
There still are. They're probably in your house. Yeah. Again, that's why it pays to pay attention to the ways that bacteria make a living, because, animals respire aerobically, which they can only do because they have mitochondria, which used to be bacteria. Plants photosynthesize, which they can only do because they have originally symbiotic cyanobacteria for photosynthesis. But when you get into bacteria, many of them can respire in the absence of oxygen because they can use other oxidants, things like sulfate, uh, oxidized iron, some nitrate, as oxidants for respiration. There are 6 or 7 different kinds of photosynthesis that don't use water as a source of electrons and don't generate oxygen. There are metabolisms that only bacteria and their cousins, the archaea, can do, like chemosynthesis, that is something like photosynthesis, in which you fix CO2 into organic matter, but the energy comes from chemical reactions rather than sunlight. And so if you look back at those, again, I think there's considerable uncertainty about what early metabolisms were, but there are at least many people who believe that they did involve very early on carbon fixation via a process that's now only found in some microbes, and probably were chemosynthetic. That is, they took advantage of chemical gradients on the early Earth and used that to drive the fixation of carbon. Now, in some ways, the key metabolism to evolve was so-called oxygenic photosynthesis, which we always think of as green plant photosynthesis, although we really ought to think about it as cyanobacterial photosynthesis, because they're the only group of organisms ever to evolve it de novo. And in that one, water supplies the electrons needed to reduce CO2 to organic molecules, and oxygen is given off as a byproduct. And that, without that evolutionary event, we would never have come to have an oxygen-rich planet. So it's just maybe the primary example of Earth's physical environment having a strong dependence on the activities of organisms.
I'm not sure if the nominalization of something that undergoes chemosynthesis is a chemosynthesizer or a chemosynthete. I prefer chemosynthete, I think, for just aesthetic purposes, but I'd like to talk about them for a minute. Do we have reason to believe that there were extremophiles already early in Earth's history? I was actually pleasantly surprised to see that a Stanford researcher once proposed that places like super hot deep sea vents may have been among the only places on Earth to survive at times in Earth's early history, such as when it was being bombarded by meteors.
Yeah, I think it's reasonable to think that on a relatively short time scale, which on the time scale we're talking about could be tens of millions of years, if not hundreds of millions of years, life figured out how to exploit almost every habitable environment. Now, not every environment is habitable. It can be too hot, it can be too dry, it can have too large concentrations of salts, you name it. But there are organisms, as you mentioned, that are called extremophiles because they live, frankly, in environments that we can't. And some of those on the deep sea, where pressures are high, live at temperatures above 100 degrees
113.
Celsius. Yeah, something like that. There are ones that live in very salty environments, and so all that is a matter of natural selection, for membranes that can sustain activity under these conditions. There's a lot of things that go on, but I think the evidence, at least from the world we see today, is that life has been very versatile in its ability to evolve, forms that can be sustained in what for us are unusual environments. And again, if you turn all that around, what it really says is that humans and animals in general are actually not very good at exploiting extreme environments, whereas bacteria have done it for billions of
Hmm.
years.
Yeah, we to leave our equatorial home regions, we just the only things we really picked up were new clothes and air conditioning, and that hasn't gotten us that far. Unlike tardigrades, which can, my understanding is they can persist pretty much everywhere, and they don't need new clothes or air conditioning for it. But
Yeah, but I'd rather be a human,
me too,
to be
me
honest.
too. You already referenced comparative biology, which I expect will fit in here, but how, when you examine a microfossil, are you able to tell whether it's photosynthetic or carnivorous or anything else about what it did? Because you can't observe it the way that we might a living creature in a lab or a cat in your lap. And as far as my understanding of fossils goes, the creature's original constituents, at least for macroscopic objects, macroscopic creatures, are gradually replaced by minerals and other deposits. So you're not necessarily even working with the original material. So is there just a close enough resemblance to living creatures that you're able to generate credible hypotheses about how they functioned?
Yeah, I mean, largely what we have in the geologic record is morphology, whether that's, a human bone or a clamshell or a trilobite, you name it. And, for those things that are fairly closely related to the living biota, you can say a great deal about them. So if you give me a clamshell that's 100 million years old, I can probably tell you a lot about it because I can compare it to living things that are similar and make some quite reasonable deductions. It gets harder as you go back, deeper in time to things that don't have close living relatives, and gets at its hardest in that 85% of Earth history that's mostly microbial. But even there, one of the great things about cyanobacteria is that not only are they probably the most important group of organisms ever to evolve because they're responsible for our oxygen-rich atmosphere, but they're different from many kinds of bacteria in they're commonly larger. They commonly have more complicated um, morphologies and division cycles, and we know a lot about how they decay, how they preserve. So there are things that I can show you that are 2 billion years old that I have very strong confidence that they were cyanobacteria that worked, at least in a broad way, like cyanobacteria do today. And then when we start getting eukaryotic life, one thing that one of my former students, Susanna Porter, found deep within the Grand Canyon in some 740 million year old rocks are about 100 micron things, sort of look like a flask that's open at one end. And we know that those are very similar to groups of protozoans today that actually eat other protozoans. And in fact, Susanna has found evidence of, probably calling it a bite mark isn't the best thing, but of punctures made by predators on these shells. So sometimes you look at it and you can't say much at all, but in other cases there is a distinct enough morphology, and sometimes you have evidence of behavior through orientation and things like this that we can use, and they tell us a lot. And then finally, there are some examples where we have molecular evidence. Recently, there was a very nice paper on these so-called Ediacaran macrofossils, which most people I think correctly, are early animals that are quite distinct from most animals living today. And it was found that they have the remnants of cholesterol in them, which is an animal-specific uh, sterol or steroid molecule. And so even under favorable circumstances, you can even get some molecular preservation that helps zero in on the nature of an organism.
Well, I would like to now step back and talk about some of the not that these haven't all been big picture ideas, but some of the bigger picture ideas that emerge from this discussion of early life before hopefully we have some time to touch on Mars. But you wrote somewhere in Life on a Young Planet that from our current vantage point, we tend to think of life as being a process of generation and replacement of species. So, in my own words, one day the polar bears are gone, and then the next, some new sort of finch is discovered on an isolated island. But on a larger scale, evolution is constantly spreading out and bringing with it new forms and ways of living. Could you expand a bit on this idea if I've captured it correctly? Because it does diverge from our intuitive picture of how evolution works.
Yeah, no, sure. I'm happy to. And I think I wrote that in response to what I sometimes call the generation of Abraham approach to the history of life, that first there were bacteria, then there were protozoa, then there were animals, then there were mammals, then there were us. And in fact, there's probably just as many bacteria now as there were 4 billion years ago, probably more. And so on the broadest scale, diversity is additive. You start out with all bacteria and archaea, then you add single-celled eukaryotes to those, but you don't get rid of the bacteria. The single-celled eukaryotes can't complete the carbon cycle and sulfur cycle without them, still can't. Then you add multicellular forms, some photosynthetic, some heterotrophic to those. And yes, on a smaller time scale, species come and go. That's still the case. But I think it is useful to step back and if you look on the broad scale of our planet's entire history, we see that there is this important component of, just increasing diversity of type, not just species.
I I guess this might not necessarily count as diversity of type, but diversity of species at least is. I read or heard at some point that they've discovered bacteria that live exclusively on the side of highways where they consume rubber from ruptured tires. And given that this wide lens that takes microscopic organisms into account as well as the macroscopic species. I mean, take COVID and its variants, for instance. It certainly does seem like they're constantly, like with the snap of a finger, spreading it out and adapting into various forms. And on longer and longer time scales, these branch out into diverse morphological types as well.
Yeah, I mean, you touch on an important point, and that is products of the human presence on Earth have created new environments that microbes have exploited, and there is now very active experimental effort to try and exploit those bacterial capacities for the benefit of humans. So, for example, there are any number of labs that are trying to find out whether we can use bacteria to break down plastics in the ocean, for example, bacteria to clean up oil spills. So in a sense, yes, it is, I think, fitting and proper to just see what remarkable evolutionary capacities bacteria have. And it is also, I think, reasonable to think that it's worth understanding those, because they, at least some of them, could be put to service for the betterment of the human condition.
I would feel much more morally comfortable enslaving hordes of bacteria to do our work than horses or other intelligent animals. Seems like an ethically free lunch to just give them a free lunch on plastic or oil in the ocean.
Yeah, it's going to happen.
Another theme that we've touched on, particularly with reference to the cyanobacteria and oxygen generation, but organisms and the environment co-evolve, as I mentioned earlier, and as I think will come up again as we talk about Mars. But is there a way of summarizing just how the atmosphere has shifted on a wide scale over the past billions of years on Earth? Like, what role do prokaryotes play in all of this beyond maybe just oxygen? What have the general shifts been?
Right. So the first thing to know is that the only source of oxygen on this planet that is sufficiently large to oxygenate the atmosphere and oceans is photosynthesis. And so the amount of photosynthesis that goes on depends on nutrient availability, which itself is related to physical processes on Earth. And also, if I have a photosynthetic organism here that takes CO2 and water and makes organic matter and oxygen, and then I have a respiring heterotroph like you and me over here that takes organic matter and oxygen and respires it back to CO2 and water, that's a zero-sum game. And so the way you break that zero sum is, again, through geologic processes where organic matter produced by photosynthesis, either directly or indirectly, gets buried in sediments. And then that creates the possibility that oxygen will increase in the environment. So what that tells us is that the process of photosynthesis, the nutrient availability that tells us how much photosynthesis is possible and who's doing it, a lot of that's coming from the biological side, but there's important components of the availability of nutrients, the burial of organic matter that come from the physical side. And when we put those together, the pattern, at least that's emerged, is that for the first 2 billion years of Earth history, there seems to be, at best, very low and transient oxygen in Earth's surface environments. Then about 2.4 to 2.2 billion years ago, there's a revolution. And now the atmosphere has some oxygen, the surface ocean does, although most of the deeper ocean remains oxygen-free. And that certainly required cyanobacteria. It also happens at a time when continents are emerging above sea level, and so we have more weathering and erosion of nutrients into the ocean. So I think it probably represents an interplay of Earth and life. The amount of oxygen that accumulated actually wasn't that much, maybe 1 or a couple % of today's levels. And that may have fluctuated a bit through time, but it doesn't start going up permanently until, around 600 million years ago, and that's when chemical signatures tell us oxygen is starting to go up. We see a fundamental change in Earth's phosphorus cycle at that point, which means there's more nutrients becoming available. We have evidence of that oxygen is going up, and that's on the timescale we start to see macroscopic animals, which need relatively high oxygen concentrations to function. And again, that still didn't bring us to modern levels, probably something like 10 or 15% of today's levels. But then by about 400 million years or so ago, during the Paleozoic era, so-called, we have evidence that oxygen is going up to essentially modern levels. And as I said, I think earlier, that coincides with the evolution of land plants, which have some decay-resistant tissues that accumulate in sediments, giving us coal and things like that. So on the long run, then it looks like you can divide Earth history into kind of 3 chapters: no oxygen, a little oxygen, and a lot of oxygen. And the boundaries between those 3 chapters do seem to represent places where, in various ways, the physical and biological Earth are starting to interact in new ways.
That gets us to exactly where I wanted to go. We've skipped a lot to reach the present, but with the exception maybe of some of the experimental research programs we've talked about with regard to figuring out the early processes the processes that led to early life and perhaps their relationship to synthetic biology. Most of our conversation has been backward looking, and far backward looking at that. But granted that your expertise in what's happened over the past billions of years of life, how does it equip you to think about what's going on in Earth's life and atmosphere today that other biologists or zoologists might not be attuned to? And I know that this is a huge subject that we don't have time to get into all the nitty gritty of, but I just like a general idea.
Yeah, well, I'd be hesitant to say that other biologists aren't attuned to this, but I do think that aspects of the past do provide a distant mirror that helps us to understand what the rest of the 21st century and beyond may be like. Some years ago, um, I got interested in what is the largest known mass extinction at the end of the Permian period, 252 million years ago. And I just started thinking about what might cause this. And I was very taken by the fact that geologists had done a very good job of documenting the facts of the extinction and some of the environmental changes, but not a very good job of relating that to who died and who survived. And it struck me that that was a little bit like going to a murder scene and not looking at the victim. Um, and so what I did was it struck me that some of the physical processes going on might have produced a lot of carbon dioxide. And we now know, we didn't at the time, but we now know that this is the time when some of the largest volcanism that we know of in the history of the planet took place. And it would have spewed immense amounts of CO2 into the atmosphere on a fairly short time scale. So what I did with my buddy Dick Bombach was literally, we went to the library for 3 months and just read the experimental literature on how do organisms respond when you subject them to high CO2 levels. And it turns out some respond very poorly, some can actually compensate for it. And it turns out that when you divide the living biota up into the things that are vulnerable and perhaps more tolerant of, increased CO2, that does an excellent job of actually predicting what did and did not go extinct at the end of the Permian period. And so I think that was probably the first time anyone ever said rapid increase in CO2 could be very bad for the biota. Well, fast forward to the 21st century, and we are now increasing the amount of carbon dioxide in the atmosphere at a geologically very rapid rate. We are seeing the effects of this on the biota, which are very similar in many ways to the effects that we see recorded at the time of the Permian extinction. And it's led to some, in my case, some, very happy collaborations that I recently co-authored a paper with a group in Germany who are probably the leading lab looking at the physiological response to elevated CO2 among marine animals. And I think it shows that, the past does have something to tell us about the future. It certainly is consistent with everything we're observing today, and it suggests that unless we are smart enough to change our ways, that we will move toward what we are moving and have already moved significantly toward a warmer Earth. That is going to have consequences for different types of plant and animal. And importantly, it will have consequences for humans as the capacity of different areas for agriculture will change, water availability will change. So we're heading into an unknown future, but in some ways, what the past tells us is that we should think very hard about how we collectively can respond to what we have done industrially, and all of us contribute to it, but how we can try to rein in global change in a way that will, vouchsafe happy, habitable environments for our grandchildren.
I've had to work on my ability to generate natural transitions. And my natural transition is, we've now learned about your wealth of experience in the past of this world, and now the future of this world. But your experience also extends to other worlds. And I understand that you served on the team of NASA's Mars exploitation or exploration probably
Not
probably
exploitation.
exploration rover MER mission. And given that this mission ended about 5 years ago, maybe before we get into your role, could you just lay out the basics of what the mission consisted in, the non-exploitative explorative mission?
Yeah, this was the first of a series of rover missions, which, there are two others still operating today. It was not to find evidence of life, either ancient or modern, on Mars, but to understand Martian environments, past and present. And in essence, the rovers, their names were Opportunity and Spirit and Opportunity, which might ring bells with some of your viewers. And in essence, these rovers were doing much the kind of work that I've done on Earth in the past. They look at ancient Martian rocks. There were chemical instruments that could tell us something about the chemistry and mineralogy of these rocks. There's even a hand lens, so you could see very fine scale textural features of the rocks, a series of cameras. And so really for about 13 years, Opportunity in particular was able to traverse among outcrops of ancient rocks and tell us something about the history. It's our first real ability to try and reconstruct the history of another planet, and it was really a privilege to be part of that team.
Yeah, and what was the part you played? What was your role in the project?
Well, too, I think one of the reasons why I was asked to join the team was simply that I did have this experience of trying to understand environmental history from ancient rock. So along with all the other team members, I was involved in interpreting what the rovers told us on a daily basis. Also, I was one of several people over the course of the mission who was responsible for what's called long-term planning. And to think about that, you have to make both tactical and strategic decisions. Tactical decision is, what should we do tomorrow? Strategic decision is, what are the big questions we can ask of this outcrop, and how do we go about answering them? And so I was one of the long-term planning or strategic leads as well. But it was very much the nice thing about the mission was that everyone's voice was important. We all listened to each other. We learned how to integrate the results from different types of observations, and because of that, we were able to learn something about what Mars was like as a young planet.
But you made it clear at the outset of your response that this project was not so much or not at all about astrobiology, but was really about geology. And this notwithstanding, did you determine whether or not the soil or terrain more generally was conducive to life? And I suppose that in asking this, there are two questions. One, do you think it is possible that there might have been life, at least as we know life, on Earth in Mars past? And in a more forward-looking sense, could we, in theory, transport any of Earth's own microbes there and expect them to thrive?
There's a number of questions in there. Let me try and unpack it. Yes, the mission had no instruments that would be specifically designed to detect or characterize life. the idea was to say, what were environments like? Because if the environment is not conducive to life, then the story is over. Now, some of the more recent missions have more sophisticated instrumentation that can help, although I'm not sure they can or have reported anything that is definitive evidence of life past or presence on Mars. But what we were trying to do was to understand the planetary history, which then provides a framework for thinking about some possible history of life. And before we ever landed in Meridiani Planum for Opportunity, people had known from remotely sensed observations by satellites that Mars was once wetter than it is now. There's ancient river systems, there are ancient deltas, lots of things that tell us that in the past there was liquid water on Mars. Now, the rocks that we looked at on Opportunity, where there are signs of water, but as I like to say, it's signs of water waving goodbye. So we have certain mineralogies and features that could only have formed in the presence of water, but they would not persist they would persist only in the absence of water. So certain types of minerals form when, they need water to form, but if you keep water around, they will transform into other minerals. And so what we found, and my former postdoc, Nick Tosca, who's now a professor at the University of Cambridge, were able to determine that, in the places we were looking at, Mars went from, at one moment, being wet environments, but as they dried out, you basically left the habitable zone of life, and there's very little evidence that they ever got wet again. So, and I think other places have seen this, that there's evidence that water could have been present for tens of thousands of years in some places. There were lakes, there were deltas, that sort of thing. But, you spread that out over a period of 1 billion years, and it could have been dry much of the time. And, some people, myself included, but not everyone, is of the opinion that while Mars was at some times relatively wet, that early Mars was episodically wet and dry. And so it's the implications for life are very different if you think that Mars was wet for 1 billion years versus Mars was wet for 100,000 years, then dry for 1 million, then wet for 10,000, then dry for another 2 million. And we're still struggling to understand the details of that. But I think it's at least reasonable to think that even in the earlier history of Mars, which clearly has evidence for running and standing water, that those might not have been continuously present. So at this point, we don't know whether life ever began on Mars. If it did, it didn't persist, at least at the planetary surface. I think if you just drop random bacteria onto Mars, which we've probably done through some of the rovers, they would not be likely to persevere because there is so little water, so little access to nutrients. So I think if you want to have life on Mars, you got to get Matt Damon to go up there and grow potatoes in some human-built structure. I'm skeptical of the idea that Mars is currently not only an inhabited planet, but at the surface at least a habitable planet.
Would you add the caveat that you're skeptical that at the surface it is a habitable planet, but only if Martian life resembles Earth life in the sense that it requires water, because presumably there are myriad other possibilities that life could take that where water isn't so vital?
Well, that's an interesting question, and a lot of people have thought about it. I think we, in some ways, instinctively think about life as we know it, which is equivalent to life on Earth. As I used to say to my students, that if you don't make that assumption, games without rules have many possible outcomes, and so you can just speculate all over the place. Now, having said that, water is a very unusual substance in many ways, and it's a particularly bio-friendly substance. And so while there might be places in other types of planets where other molecules could play the role that water does, on most rocky planets, including Mars, I think water is probably the only game in town. And there are certain characteristics, physical characteristics of water that are likely to affect life of any sort. On this planet, once you get to a salinity above a certain point, life doesn't exist. And it's the reason that our ancestors used to salt meat or add sugar to preserve fruits. And that lowers the so-called water activity, that is the percentage of water molecules that at any given moment can participate in the chemistry of water. And I think likely the limits imposed by water activity, the limits imposed by something called chaotropy, which some ions are actually destructive of polymers like peptides or nucleic acids. So I suspect that while we don't know that life on Earth is the only type of life that can exist, my guess is that wherever we find life, it'll be based on carbon. There's a fairly large chance it will be environmentally based on water. And that's about what you can say.
That's a very helpful answer. Yeah, okay. That's great. I've spoken to one other astrobiologist. I don't know if you want to consider yourself an astrobiologist, but I've spoken to one other, I'll call you one. I've spoken to one other astrobiologist on the show, and her name's Clara Sousa Silva. I think she was at Harvard for a while. She's at Bard now, but she was part of this project, I believe, led by Sarah Seeger of MIT, that you might recall this a few years ago. They believed they'd found a signature of life on Venus, particularly in the clouds of Venus. But the way that Clara works is by looking at the spectra of exoplanets to determine the contents of their atmospheres. And based on these contents, she's then able to deduce whether there's a possibility of life there, because some atmospheres, particularly atmospheres in her case that are high in a compound called phosphine, are highly unlikely without life because they require a particularly complex chemistry that you are unlikely to find in geologic features. But as you know, as we've already talked about, life has very much created the atmosphere that we now inhabit on Earth, which is why finding phosphine would be so important. But is there, in the same way that I sort of simplistically laid out how her astrophysics and chemistry work functions in an astrobiological context, is there a simple way of describing the way that your more terrestrially focused work would become applicable to astrobiology in the event of a mission that were explicitly devoted to astrobiology? So, for instance, what sorts of things on a rover like Spirit and Opportunity would you want it to have so that it would be optimized to search for life when you're getting data sent back to you?
Yeah, there's a series of good questions in there.
I'd like to hear that.
I think
Thank you.
I think the first thing to say is that the way you can do astrobiology depends on what you're looking at. The kind of work that I've been privileged to do is something we can do in our own solar system, because you can go there and at least send a rover, if not a human being. And I think over the next 20 years, we're going to see missions to moons of Jupiter and Saturn that are candidates for biological planets. The kind of work you were talking about on looking at planetary atmospheres, that's what we can do to looking at extrasolar planets, at least within our own cosmic vicinity, a couple of light years. And people like Sarah Seeger and others have made real progress in our ability to actually understand the chemical composition of other atmospheres. And that's all exciting work. One of the questions, which provokes a lot of debate, is whether there is any atmospheric signature that is uniquely and diagnostically biological. And I have to tell an anecdote here, because years ago, I was having dinner with a man named Dave Stevenson, who's a Caltech professor, one of the great planetary scientists of our time. And we were just talking about this subject. And, I sort of parroted what everybody else was saying at the time, and still are largely, that, if you found an atmosphere that simultaneously had oxygen and methane, since those two don't coexist at equilibrium, that would be evidence of life. And Dave just smiled and he says, I'm willing to bet you that when that observation is made, within a year, there will be at least 6 published papers showing models of how that can happen without invoking life. And I think that's certainly what happened on a more rapid time scale with the phosphine observation. And so I think it remains an interesting question that deserves careful thought about, what can you say definitively about habitability or inhabitation based on a planetary atmosphere. And then, if you think about it, for most of the universe, the only way we'll ever know if there's life out there is if they tell us. That's really where that comes in. So I think we're going to see more bio-focused instrumentation on different missions. I know there's a lot of people who would like to go to Enceladus, moon of Saturn, that certainly has geysers. And those geysers are known to have organic molecules in them. And so you can take instruments that would gather and analyze the composition of those geyser effluvia. Going back to Mars, there are more sophisticated ways of examining organic matter. My own feeling is that what we would love to do, and I know it's technologically challenging, would be to actually drill relatively deep holes into the Mars subsurface, where not only is there some possibility that there is a subsurface biota, but more importantly, unlike surficial materials, things at depth might have had less alteration due to interactions with the surface environment. So there's a lot of things that will happen. It is, putting together a mission to any of these places is really challenging. And I admire the people who do these missions and carry them out. But my guess is that over the next 20 years, we will see both increasingly sophisticated and biologically oriented missions within our own solar system and an increasingly sophisticated efforts to understand planets beyond our solar system based on chemical signatures that we can see by remote sensing.
Well, Andy, this has been all around terrific in so many ways. We only touched on a small fraction of your work. I mean, there's a lot that happened between that early life and where we are today. But I am so thankful for your time. This was a great start on this subject for the show, and there's room for so much more about it. So again, thanks for your time. This was really great.
Thank you. It was fun.
Hold on. If you haven't subscribed, liked, commented, or reviewed, that would be so helpful. And if you haven't yet, you could also follow me on Twitter and Instagram at Robinson Erhardt.